Hi, hello. Welcome to the Physionic podcast, which is going to be a bit more of a lecture this time around. This is actually a lecture that I gave just maybe a week ago, 2 weeks ago, something like that, for an undergraduate class. So it spanned over 2 days, and it was a lecture on muscle, really honing in on skeletal muscle, but I'll also just touch on smooth muscle and cardiac muscle as well. So, if you'd like to, essentially, if you don't have any background in muscle, or if you don't have any background in physiology and you want kind of an introduction to how muscles work, where they're located, and that's not as obvious as you might think, as well as metabolism and how they're activated and things of that nature, kind of from the ground up, then this is a perfect lecture for you. It's not too involved. I cut out a lot of the details that I just don't find are that necessary. Going through a lot of muscle lectures myself, through my master's degree in exercise physiology, and certainly during my PhD, I feel like a lot of lectures end up getting bogged down with a lot of the minutia that simply I've just never found that it mattered. So this is going to be a relatively high level, but still a college level course that or class that I'll be teaching on the podcast. So I'm going to have a ton of visuals, of course, because it's going to be a slide presentation, and hopefully you get something out of it, because I do think I've put a lot of work and thought into how I'm going to present this, so hopefully it comes across. For anybody that's new to Physionic and doesn't know who I am, my name's Nicholas Verhoeven. I'm a doctoral candidate in molecular medicine and hold my master's in exercise physiology. So that's why I ended up teaching this muscle lecture. So I thought, why not spread the wealth and offer that to the Physionic subscribers as well. So I'm going to be breaking this up into two sections. So the first part is going to be today on this podcast, and then the second part, which is going to be, the first part is covering a bunch of the basics of muscle, and then the second part is going to be touching on the metabolism aspect, as well as some of the types of different muscle cells that we have, as well as potentially, if I decide to throw it in there, depending on if you're interested, I'll also throw in a clinical mystery that I created, which is going to be based off of all the information. So you won't have to know any added information other than what we cover in this podcast and the next podcast. And at the end of the next podcast, then I will have this clinical mystery for you to solve. And it's not overly involved, but I think it's kind of cool, and it kind of stretches your mind and your mind muscle in a way, even though your mind isn't made up of muscle. Okay, but without further ado, let's go ahead and jump into this, and hopefully you learn a thing or two from this lecture. So I've creatively called this the muscle lecture, which is rather apt considering that's what we're going to be discussing. And I've got a few questions in here, kind of strewn in here, because I like to have an interactive classroom. Obviously, online, it's a little difficult to do. Maybe with the online chat, I can do that a little bit, but I'm probably just going to be blasting through these. But as you're going through this lecture with me, just think about some of these questions that I'm going to be posing. And hopefully, some of the answers are super obvious. So don't think that just because if you don't know an answer, you think that, oh, surely it's not this obvious. No, it sometimes truly is this obvious. So the first question is kind of, why do we have muscle? And some of the obvious answers, or some of the answers that I got during the lecture, were movement, right? So just to be able to move, for me to be able to move my mouth and be able to speak, and I tend to be kind of, I gesture a lot with my hands. I suppose that's maybe my European background, but that is all powered by muscle. And beyond that, my ability to blink my eyes is another form of movement, and it's another thing that's powered by muscle. But even beyond that, there are other areas of musculature that we don't actually think about beyond picking things up and movement and whatnot. So I'm going to go ahead and kind of detail some of those. So the first one, as I mentioned, was movement. And That can be from something as simple as writing, with a pen, to typing, to eye movement, blinking, and to the really obvious of running or jogging or walking or just moving your hands around. The second one, which is far less obvious, unless, of course, you've got a little bit of a background to this, is that your absorption is actually based off of musculature as well. This particular type of musculature is called smooth muscle, and it works differently than skeletal muscle, which is skeletal muscle is what I'll be focusing on. But smooth muscle, for example, with the absorption, with the ability to absorb different nutrients that you consume. So let's say you consume a large amount of food. When that enters your intestinal tract is aligned with smooth muscle cells specifically. And these cells will squeeze together. And that whole process is known as peristalsis, where they're moving your food from the top part of your intestines, so right around here, and it moves all the way through all of the small intestine, the duodenum, the jejunum, the ileum, and eventually ends up in the colon or the large intestine. And all of that is powered by these muscle cells that are constantly contracting back and forth to allow for that movement. Another form of musculature is cardiac muscle, which is probably a pretty obvious one as well, that your heart is primarily made up of muscle. It's got this sheath known as myocardium, and it is made up of cardiac cells, which look different and act different from skeletal muscles as well as smooth muscle, but they're definitely more akin to skeletal muscle. So, just think about that in a really obvious sense. You have to have movement, right? You have to have the contraction of the heart to end up squeezing blood throughout your entire system. But beyond that, actually, not just your heart. The heart is probably the most obvious one, but your entire vasculature. And I've discussed this on Physionic many times in some of my videos, that with the cardiovascular system, the vasculature, the actual circulatory system itself, like your arteries, have linings of muscle cells around them. Again, in this situation, we're talking smooth muscle. And these cells, again, squeeze in or relax. And that's when they squeeze in, they increase the pressure in the arteries. And when they relax, they open up those arteries so that you can decrease your blood pressure, which is obviously necessary if you have too high blood pressure in certain situations. So the point is here, though, that muscle is all over the body and has all kinds of different functions. So if we were to take these cells taken out of these different regions of the body and put them on a plate, on a petri dish, or really a cell culture plate, which is what researchers do, it's what I've done in the past, then you'll notice that the shape, or what's known as the morphology, of these muscle cells is quite different from one another. So you've got your cardiac cells here, you have your skeletal muscles, which are kind of longer, tend to be more tubular, and then you have your smooth muscle cells, which their actual structures really quite different as well. And this is all just in a cartoon form, but I'll be showing you some real images of what this stuff actually looks like going forward. So the point is that all of this stuff looks very different, has ultimately the end function is the exact same, but they look different and achieve that function in slightly different ways. So really, I'm going to be focusing on skeletal muscle, like I mentioned. So let's imagine that we take a sample of skeletal muscle and put it onto a dish. And I'm going to take Daniel Craig as my victim here, as my participant of our study that we're going to be going through for the rest of this lecture. So we've got Daniel Craig, and obviously he has muscle as well, just like anyone else. So he's got this bone, which is the top part of his arm, as I've detailed here, and it's technically called the humerus. Not that it really matters what the bone is called, but the muscle is then attached to the bone. So because that's the only way that we can actually get ambulation. It's the only way that we can actually move our body back and forth. So with this muscle belly, which is the entire musculature itself, is bound or hooked up to the bone at two different sites, usually two different sites. It can definitely vary, but two different sites. In this situation, you have it at the top of the arm, so where your shoulder is, and then another connection at the elbow. And clearly, this, if you're looking at the back of the arm, you're talking about your tricep muscles. And if you're looking at the front of your arm, you're looking at your bicep muscles. I think in this situation, I created more of tricep muscles, but it doesn't really matter. Either way. So if we were to take that muscle out and remove it from the bone and just look at the muscle belly itself, we notice again that there's the muscle and then the tendon. Now within that muscle belly, however, are muscle cells, because just like anything in your body, everything of your body is made up of, well, maybe not everything, but the vast majority of the things that make up your body are cells. And they come in different shapes and sizes and functions and do all kinds of crazy stuff. But specifically in this situation, we're talking about skeletal muscle cells as opposed to smooth muscle or the cardiac muscle I was talking about earlier. These skeletal muscle cells tend to be longer, tend to be more tubular than what you would see from some of the other muscle cells. So if we take those muscle cells, a grouping of muscle cells, and then we zoom into one muscle cell, now we're down to our single muscle cell, and we were to take away the sheath that is the cell membrane around this muscle cell, we'll see that there's a ton of different components that make up that muscle cell. And muscle cells are also interchangeable with terms like myofiber or myocyte. That's another. So there's a variety of different names. But the point is that if you take a single muscle cell, and here in the image at least, we've got this sheath that goes over the entire muscle cell. And here they've cut away in this diagram, they've cut away the actual cell membrane so that you can actually see some of the inner workings within. And I've got these things kind of labeled off. So 1, 2, 3, and 4. So number 2 would be your cell membrane. Number 1 is the powerhouse of the cell. I'm not even going to tell you what that is because I'm sure you know from your biology classes. Number 3 is your nucleus, and something really unique or relatively unique about skeletal muscles is that they have multiple nuclei. Nuclei being these organelles or these sections of the cells that contain our genes. And with the containing of those genes, then we can generate more proteins and allow for those muscle cells to grow. I know I've talked about satellite cells and the donation of nuclei and all that stuff in other content as well. So it's just a big carryover here. And then number 4 is these myofibrils, which we'll go into as well. So just to uncover these, you got the nucleus, sarcolemma, which is the same as the cell membrane. Muscle biologists like to have their fancy terms as well. So that's why they end up complicating things by having their own names. So sarcolemma, then mitochondria, the powerhouse of the cell, and myofibrils themselves, which are components within the muscle cell. So if we take a camera now, if you're looking at the podcast, you can see that I've added a little camera here, and it's looking into one of these holes that seems to be inside the sarcolemma. So it's a hole that goes into the sarcolemma. So what's going on there? Well, it's not that your muscle cells are actually riddled with holes, even though it looks like it from at first glance. But if you take that camera and you look inside, you see that there are these grooves, there are these trenches that are found inside the muscle cell, and there's a particular reason for why those are there. But regardless, even with those trenches, the sarcolemma is still present and is always over the actual muscle cell. So the sarcolemma will be flat, just like any other cell, and then suddenly it'll come down into the trench itself. But that's not to say that just because there's a hole there that the inside of the cell is actually exposed to the outside environment. So this camera is looking down, and here we've got our sarcolemma, we've got our cytosol, or our sarcoplasm, if you want to look at it from a muscle biology perspective, which is all within the cell, and this white section is all outside of the cell. Okay, so here we've got I guess you could call them like a receptor. So you've got these receptors that are found inside the sarcolemma, and then you've got another receptor that's found inside of what's known the sarcoplasmic reticulum. The sarcoplasmic reticulum is a really important structure within the muscle cells. If I can back up here, this kind of purplish section here that kind of looks like a web, almost looks like a spider web, is the sarcoplasmic reticulum. And you can see that it kind of goes all over the muscle cell, all the way down. It's all over these myofibrils, and it even goes within the muscle cell quite deeply within. So why is that? Well, it contains calcium, and calcium has a really important function that is dependent on these two receptors, or these two gateways that we're going to be talking about in just a second. So calcium is found in the sarcoplasmic reticulum, and there's a lot of the sarcoplasmic reticulum that stretches across the entire musculature. And we have this inactive muscle cell, and really I'm putting that in quotations, because do we really have an inactive muscle cell ever? Not really, but in terms of how we're going to be defining things as contraction and non-contraction, this is an inactive muscle cell, so it is not contracting. Okay, so this trench that I was talking about earlier is called the T-tubule, and it allows for a signal, and we'll go into what that signal is later on, but it allows for a signal to actually travel down the sarcolemma and activate this receptor, the first receptor which is found on the sarcolemma. And that receptor is called the dihydropyridine receptor, or DH. I guess it doesn't really matter, but DHDR, DHR, I'll just go ahead and call it by its name. The dihydropyridine receptor is then mechanically linked. So there's actual molecules linking the dihydropyridine receptor to the receptor that's found on the sarcoplasmic reticulum. And they're very close to one another. So the sarcoplasmic reticulum comes right up very close to the sarcoplasmic, or to the sarcolemma, excuse me. So there's very little room between those two. And this allows for a mechanical linkage between the dihydropyridine receptor and the ryanodine receptor, which is found on the sarcoplasmic reticulum. But both of these are still closed, right? So we haven't activated anything, so the muscle cell is as quiescent as it can be. Now, whatever that stimulus is, which we'll get into, it ultimately ends up leading to the dihydropyridine receptor opening, or having a shift in that receptor, which leads to a shift in that mechanical linkage between the dihydropyridine receptor and the ryanodine receptor on the sarcoplasmic reticulum. Now that then in turn leads to an opening of the ryanodine receptor, and the calcium will then flood out of the sarcoplasmic reticulum. So it went from nicely nestled and held in this sarcoplasmic reticulum to now being out inside the cell, just all over the cell. So it gets released from inside to outside, but still within the cell. So we're not creating gaping holes in the sarcolemma. This is all within the cell. And that ultimately leads to an active muscle cell. But what exactly does that mean? Well, let's go into it. That calcium ends up interacting with a series of structural proteins that allow for muscle contraction. So really, this is called a sarcomere, but sarcomeres make up a myofibril. So and I mentioned myofibrils earlier, so let me go ahead and go back and touch on what myofibrils are. So you've got our muscle cell again here, and we look at one section, one part of that muscle cell is known as this myofibril. And it looks like another cylinder. It just looks like another muscle cell, but it's on a much, much smaller scale. And this myofibril has a certain structure to it that we'll get into it. But I did actually want to show you an actual image of what that looks like in real life, because I've been using a bunch of cartoons up to now. So here we've got this myofibril, and you don't really need to know the I band and M band and all that at the moment. But the point is that these things are incredibly small, and you've got hundreds of them that are all within a single muscle cell, and then you have to think about compounding that multiple times over and over and over until you get your entire muscle belly. So if we then really focus in on some of the structure, and this is actually going to be important for us to understand how muscles actually contract, we see that if we just look at a piece of that myofibril is made up of a series of different sarcomeres. So sarcomeres are just these end-to-end connections, and you can see them. So they're defined, quote unquote, defined by their Z disc, which is a simple line that contains a bunch of other proteins within that line. So just think of it like a square or a rectangle. I think a better way to look at it is a rectangle, and the edges of that rectangle, the long edges of that rectangle or at either end of the long edges of the rectangle, are the Z lines. And then within that, between those two lines, are a series of different proteins. And that's known as a sarcomere. So within that sarcomere, you have these darker regions, which is, again, this is a real image compared, and I'm just comparing that to the cartoon image. You've got a real image of this kind of darker section, and this darker section is defined as an A band, and that A band is made up of two particular proteins that I'm about to go into. So the point is, for now, just know that, this is just how it looks. And it's important because it gives us this definition of striated muscle. So if you were to actually look at muscle, let's take our skin off and actually look at the musculature, you'd find that the muscle has striations in it. Think of it like a tiger almost, where we've got skin over our tiger bodies. But our musculature is very striated, and that is relatively unique to skeletal muscle specifically. You do not find that, for example, with smooth muscle. So that's due to this A band, which is made up of these two particular proteins. The space between one A band and another A band is called an I band, and the M line is an anchoring protein that's right down the middle of the sarcomere, and it anchors a variety of different proteins within the sarcomere. So point is, the big picture here is just know that there's a lot of structure that's going on, and I've left out quite a bit here as well. So if we look a little deeper at this and we actually look at the proteins that make up this sarcomere, we find that there's a series of different proteins. I'm going to be mentioning 4 of them, but really there's a whole lot of them. So when you have this one protein known as myosin, which is this, in the graphic that I have here, it's kind of purplish bluish color. That normally what that does is it interacts with this, what's known as a thin filament or actin protein. And when these are interacting with one another, there is a contraction that's occurring, or there's force generation that's occurring. And if you were to compare these two images, you'd find that there's a lot more connection or contraction or binding of myosin to the actin. And here we find that there's some open area here. So that means that it's kind of inactive. That means that it's in a relaxed state, in a more relaxed state, in a really relative sense. Again, I'm keeping out a decent amount of detail here because I just don't... I've had lectures, I've had entire classes just on the interaction between myosin and actin. Think about that. Talking about that one thing for 1 hour, it gets pretty involved. But the point is that the more interaction between myosin and actin, typically the more contraction that you have going on. So here's, again, more of an image of, and I probably should have placed this before, but you have your myosin protein down the middle, and then the actin protein that ends up interacting with it. And the myosin is also bound or interacts with a protein known as titin, which is one of the largest proteins in the human body. And that titin is sort of like a spring. It moves, it kind of stretches out, and it can also contract quite a bit as well. So this interaction between myosin and actin allows for this titin to bend inwards or to move outwards. And actin is anchored or kind of structurally put in place by another protein known as nebulin, but I don't have that imaged. So, but the big two that I definitely would want you to know, and for anybody who wants to learn basic muscle biology, you have to know what myosin is and you have to know what actin is. Those are the big two, because those are the two that are going to interact, and those are the two that are going to end up leading to muscle contraction and force generation for you to be able to lift things and do everything else that we talked about earlier. So with that in mind, we have to look a little bit closer. So we're zooming in. We're just continuously zooming in. We went from the muscle belly, we went to the muscle cells, then looked at a single muscle cell, then looked at the myofibrils, and then we're looking at a single sarcomere that makes up that myofibril. And here, then, if we zoom into this interaction between actin, which is right here, and myosin, which I've tried to color code as well, you'll find that actin has two particular proteins that are bound to it or interact with it as well. And one of them is called tropomyosin, which is kind of coiled around the actin thin filament. So if you think of actin as kind of like a rope-like structure, then this tropomyosin is wrapped around it. And the reason for that is because tropomyosin actually inhibits or stops myosin from binding to actin. So tropomyosin wraps around actin and inhibits or stops myosin from interacting with actin. So that stops muscle contraction then. So it disallows muscle contraction. So, and then you've got this other protein that's bound to tropomyosin, which is known as troponin. So let's look at what that actually does. Why are those there? Why is troponin there, for example? I'm going to have to switch diagrams, so we're going to have to leave the color coding behind because I found this diagram that I thought was far more applicable, made a little bit more sense than what I could draw out. So you've got your actin here. You still have your tropomyosin wrapped around. You still have your troponin protein, but the specific effect of troponin is that this calcium that is released leads to a binding of calcium to troponin, which then shifts the tropomyosin protein, the snake-like structure of tropomyosin, then shifts over, excuse me, so that now there's a binding site for myosin to then attach. So the way it functions is calcium binds to troponin. Troponin that is already bound to tropomyosin will then lead to a molecular shift where the tropomyosin will shift over, and this then leads myosin to have an opening to then interact with actin. So think of tropomyosin as the evil stepmother, and or you know what? I'll change the story. Tropomyosin is the stepfather, and troponin is the mother, and calcium is something else that allows the mother to convince the stepfather to step away from blocking the love between actin and myosin. And then myosin can then bind to actin, and they can hug it out. So that's sort of kind of a really classical understanding of how this all works. But really, one of the questions that I have for you, if you're watching or listening in general, is where does the calcium originate? We've actually already covered that. So if your answer is the sarcoplasmic reticulum, you're right. That's exactly right. So all of this kind of feeds back to what we were talking about earlier, that this calcium release from the sarcoplasmic reticulum leads to an interaction with the troponin, and then all this occurs. So what we have to get even more granular. So myosin is going to bind to actin, and what happens then, right? It's not just going to bind and then just sit there. It has to do something. So here's where we have this this theory known as, or this kind of factoid known as cross-bridge cycling. So cross-bridge cycling is the process by which myosin interacts with actin. And it goes through a 4-step process. And I'll explain it, but you certainly I'm going to tell you the 2 things that are absolutely necessary for you to know. So the first step is that the myosin head specifically, so just a part of the myosin, will bind to the actin molecule, the actin rope, actin thin filament, however you want to define it. So this myosin head will bind, and attached to it, with this myosin head, there's an ADP molecule, which is a low energy molecule. So think of ATP, right? Adenosine triphosphate, that I've discussed many times before. Well, this is a lowered version of that. So it's the little brother of ATP, and a phosphate molecule. So both of those are still attached to the myosin head as the myosin head comes directly up and touches and grabs onto the actin molecule. Then you have what's known as a power stroke. So this is where the actin head will snap in a particular direction, and this is facilitated by the release of this ADP molecule and a phosphate molecule. And that happens, so let me quick cut. So if you have that, right, if myosin binds in step 1, and then step 2, it cocks a particular direction. If you have that happen billions of times over, so you have billions of myosin heads that are attaching and then doing the sliding filament, or the power stroke specifically, that then leads to the entire sarcomere and the whole myofibril and the entire cell, if you just keep expounding upwards, to then contract, because that means that filament is going to start moving in or start moving out. In this situation, I'm just looking at it classically as moving inwards. So contraction, a concentric contraction occurs. So on a really simplistic standpoint, you have this power stroke that occurs from one myosin binding to actin. And you'd think, well, who cares? But again, this is occurring across billions of them. So it actually does matter, because all of your movement is dependent on this power stroke occurring. Okay, so it's done this power stroke. So now it's in this conformation, or it's in this angle where it's kind of bent over, and it needs to be reset from back to its kind of more linear, more upright position. So what happens is you have ATP, or cellular energy, that comes in, and it binds to the myosin head. And this actually allows for the dissociation of myosin from actin. So now the actin is no longer bound to myosin. And then you get the final step is what's known as ATP hydrolysis, where you get a reaction where ATP is split into back to ADP, so it's little brother that I mentioned earlier, and a phosphate. And that leads to the recocking, or getting the myosin head to be upright once again. And then you start over again. You start at step 1. But that's a lot of information, and I've gone through so many classes at this point that I'm well aware that none of this stuff matters except for 2 steps. 2 steps are really crucial, even though your professors are going to tell you that, oh, please remember every single step of this and all that. It just, unless you're a muscle biologist studying exactly this, it just doesn't matter that much. So really the 2 steps that I would really keep in mind is that you have a power stroke that actually enables the contraction itself. So that allows for your muscles to contract. And the second one is that all of this process costs energy. So this is not a free process. So when you're moving, physical activity, right? If you think about it, like where does the energy, the metabolism, remember metabolism is, in a generic sense, is basal metabolism, physical activity, and the thermic effect of food. 3 things. Where do you think the physical activity comes from? This right here. This is where the ATP comes in and to dissociate the myosin head. So you have to have energy, and this is where you're getting it. This is one of the main mechanisms where physical activity leads to the use of energy. Okay, so if you were in a classroom and I were to ask you which one out of these images is contracting more, so which one is likely producing the most force, what you should be looking for in this situation is the amount of space, essentially. Not necessarily, it's a different question to the potential to contract more. The potential to contract more would be this top one up here, because there is more space for the myosin to interact with the actin proteins. However, in this situation, assuming that you have myosin binding completely to actin, then you have more contraction or more force generation at that moment here, because you have less space here and you have more of this overlap between myosin and actin. Okay, but how do you stop this process? This can't go on forever, otherwise you're going to be just contracting nonstop. Every single muscle in your body is going to be fidgeting and twitching all the time, and it probably wouldn't even be able to twitch. It would just be continuously contracting. So that's a problem. Fortunately, your cells have systems in place where they take advantage of biochemical processes. And in this situation, there is the use of a pump. And this pump, just like pumping water, pumps calcium. So instead of pumping water out of a boat, you're pumping calcium back into the sarcoplasmic reticulum. So as your cell is doing this, that means that the concentration of calcium is starting to decrease inside the cell. And ultimately, these calcium ions that are bound to the actin, specifically to the troponin, right? Troponin activates tropomyosin, which then allows myosin to bind to actin. Those calcium ions will then dissociate from the troponin and be sucked back up into the sarcoplasmic reticulum through a pump known as the SERCA, or sarcoplasmic endoplasmic reticulum calcium ATPase. Let's just go with SERCA. So SERCA is an ATPase, just like the myosin head has an ATPase, which means that it uses energy as well. So this is also an energy-intensive process that you could probably fall into this whole physical activity aspect of metabolism. So once the calcium is back into the sarcoplasmic reticulum, ta-da, you are no longer contracting. So that's how it stops. And there's certainly more complexity, as always. Okay, but how do your muscles actually know to contract? We just went through the entire process of how they contract, but how do they know to contract? What is the stimulus for that? Well, the stimulus is these motor units. So the motor unit is defined as one neuron or one nerve cell that extends from your spinal cord and will then interact with a multitude of different cells, multitude of different muscle cells specifically. So it's not just one neuron to one muscle cell, it is one neuron to many muscle cells. So unfortunately, the neuron is polyamorous, but the muscle cells are dedicated to one neuron. I don't know, maybe they get the short end of the stick, but either way, that's how it works, that you have one motor unit or one neuron that interacts with a series of different muscle cells. So what does that look like? If you're interested in actually looking at a real image of that, I have one here where you have this neuron that branches off to all of these individual muscle cells. And you can see the interaction point. And I don't want to call it a physical interaction, but it's a point of very close proximity to one another. So it's pretty cool stuff to look under a microscope and be able to see some of this stuff. Okay, so what does that actually look like? If we go down the entire neuron and we look at the very end of that neuron, so that's known as the axon terminal, because this branch point is called an axon. So the axon terminal, the very end of it, kind of like an airport terminal, is where we get this, again, quote unquote, interaction between the muscle cell and the neuron itself. So here we've got our sarcolemma. So this is all part of the muscle cell. And here we've got our neuron and the axon terminal. So what happens? And this seems like a bit of complexity here, but don't worry, it's actually quite simple. Oh, I forgot to mention, I actually made a mistake on this image. So if you happen to catch the mistake, please comment. I'd be kudos to you if you can figure it out. If you've taken chemistry, you probably know what the mistake is. Okay, so here we've got our action potential, which is known, let's just call it an activation of the neuron. I'm not going to go into action potentials and depolarization, hyperpolarization, and all that stuff, because again, I'm trying to keep things as relatively simple as possible. We're already covering a ton of information. So the action potential, or the activation of the neuron, the activation goes down the neuron, and here then we have these vesicles, which are really just these globular circles or sacs that contain acetylcholine, which is a neurotransmitter. It's a molecule. And it's all found within the neuron, the very end of the neuron. And when the neuron is activated through this action potential, the vesicle will fuse with the cell membrane of the neuron. And when that happens, it releases the acetylcholine into what's known as the synaptic cleft, which is the space, because there's no physical interaction, the space between the neuron cell membrane and the muscle cell membrane, known as the sarcolemma. So here, once the acetylcholine is in this space, what it does is it binds to receptors on the muscle cell membrane, the sarcolemma, and binds to receptors that are known as acetylcholine receptors. This then opens up these acetylcholine receptors to allow the entrance of sodium. So sodium that is outside of the cell in high concentration relative to the inside of the cell will then come into the cell. This then leads to an increase in the what's known as the membrane potential. You don't really need to know that beyond the fact that if the membrane potential goes up, that means that there's going to be an activation of the muscle cell. So this muscle cell activation, known as an action potential, will then go down the entire muscle cell once you have this opening of these receptors. So if I were to merge the two images that I created earlier, here we've got our neuron. We still have the input of the acetylcholine binding to the acetylcholine receptor. Then you have the sodium entering the muscle cell, and then you have this activation of the muscle cell, and it goes all the way down, and you have a series of other receptors that are being opened up, or gateways that are being opened up, known as voltage gated channels. And this is the catalyst, or this is the reason why the dihydropyridine receptor that we talked about earlier then opens up, and this mechanical linkage then shifts so that the ryanodine receptors then opened. It's kind of being yanked open, and the calcium floods outwards. And that's how we activate our muscle cells. So it's through neurons. You may not be consciously thinking about it, but your neurons are constantly firing and activating to allow all of this to happen. You have to think, I mean, on a cellular scale, this is incredibly complex. But I mean, beyond that, it's also just to think that all this stuff is happening as you're listening, as you're watching. This is happening millions and millions of times over, and you have no knowledge of it. It's going on. It's just happening because your body is just that incredible. That's truly humbling. It's an incredible thing to think about. Okay, so if you were a researcher and you were to put a probe, so just a metal instrument that you're just going to touch the neuron, and then you were going to take another probe and touch the muscle cell itself, and you wanted to measure the electrical activity or the amount of activation of each, you would get these readings that I'm showing here. So I'm going to color code them in just a little bit, but before I mess up this entire image again, let me walk you through. So you have this probe that is attached to the axon or the neuron, and you get this reading of this, just completely quiet, everything's fine. This is known as the resting membrane potential. And then suddenly it just goes way up, and that's the activation. That's called an action potential. So this is an action potential, and then it goes back down. Technically, we have hyperpolarization. Then we are back to our resting membrane potential. But the point is that you have this activation. That's really the big thing I'm trying to get across. You get an activation, and then what happens, right? You have this, keep in mind, you have the acetylcholine that's released, the binding, the sodium coming in, all of that stuff. And once the sodium comes in, you have the activation of the muscle cell. So the muscle cell has its resting potential, and boom, the acetylcholine hits, the sodium comes in, and bam, you get this activation of the muscle fiber or the muscle cell. And you can tell that I just want you to look at the time here. So the time for the electrical signal of the neuron and the time for the electrical signal of the muscle are really close, but you can tell that the neuron is just a little bit before the muscle cell. And that makes sense, right? Because the signal is coming down the neuron first, then there's a chemical signal, and then there's the electrical signal that goes into the muscle cell. And then if you look at the actual contraction, so the actual, and here I'm going to color code things. If you actually look at the contraction, you see that the contraction is much later than the electrical signal, because an electrical signal is, of course, going to be faster than a mechanical signal. So this is where you get force generation. This is where you get the actin and myosin binding. But relatively speaking, this is super, super quick, and yet the contraction occurs 10 to 100 milliseconds. It doesn't take long at all, but relatively speaking to the actual electrical signal, it's certainly much slower. So you have electrical in the neuron, then electrical in the muscle, and then you have the actual mechanical contraction that occurs. So pretty amazing stuff. And again, keep in mind, this is happening, throughout the day, trillions of times. It's staggering. Okay, so we talked about muscle contraction. We talked about how your muscles know when to contract, which was through the neurons, through the motor unit. But how do your muscles know to be graceful? And I think this might be the last section that I'll be covering before I'll leave the rest for next time. So how do you get the difference between sitting down, like I am right now, and where you might be as well, and going full force for a punch or to lift weights or to lift 315 pounds or whatever it might be? Whatever ends up being, using a lot of force suddenly. How do you get that difference? How is how are your cells, how is your body able to create a gradation? Well, we have to return back to our motor units. So here I've got this multiplied by 2 motor units. So we've got motor unit 1, which comes down and interacts with its multiple muscle cells. And then you've got motor unit 2, which interacts with other multiple muscle cells. And here, the big difference that I want you to be able to tell is that the color is different. One of them is a lighter color and the other one is a reddish color or a darker color. So what causes that? Well, that is based off of a particular protein known as myoglobin that's found inside of these cells. So the more myoglobin that you have, the more of a reddish color you have. But these cells, these different types of muscle cells, have a variety of different names. You have type 1, which tend to be reddish, and you have type 2A, which tend to be kind of a cross between the type 1 reddish color and then the type 2B, or type 2X, which are kind of a lightish pink white color. So you have this almost like a gradation between the different muscle cells, and they all have their advantages and disadvantages. So this type 1 is, sure, it's red, but it tends to have a lot more mitochondria. It tends to be extremely fatigue resistant, meaning that it doesn't get tired easily. So your ability to maintain your posture, for example, is based off of these type 1 fibers. Because if you would get tired just sitting, we'd be in a lot of trouble if that were the case. But on the other hand, you need to be able to activate these other cells. And what's the advantage of the type 2B, which are on the other side, which are lighter colored? They're not just a different color. They actually produce force really quickly. But the disadvantage is that they tire a lot more easily. And then the type 2A are in the middle. They're this middle ground that kind of share some characteristics of the type 2B or X. Those are both the same, and the type 1. Okay, so let's go back to our family-friendly Daniel Craig. And we're going to be taking a muscle sample, which is something researchers do, where they take what's known as a punch biopsy, where they actually insert a needle into the arm or the muscle that they want to take a sample out of. And then they take maybe 50 milligrams of muscle. And in this situation, they're doing histology staining, which all of these kind of circular globular structures are muscle cells. But you're looking down the muscle cell. So it's like before we were looking kind of lengthwise to the muscle cell. Now we've turned it and we're actually looking into the muscle cell tube itself. And we can see that there are different colors, which is why I alluded to the type 1 and type 2A and type 2B/X. And if you had to take a guess at this, which muscle cells are type 1 and which ones are type 2, purely by the color, you might guess, hopefully you would guess, that the type 1 muscle cell is the darker colored stained muscle cells. The whitish ones are the type 2B or type 2X. And the kind of in between is the type 2A muscle cells. And this is a really basic way to look at it. So this chart explains and really shows the difference between these muscle cells. So the type 1 being low in force generation, so that's their weakness, if you want to call it that, and they're very low or very slow in their speed at which they can produce that force. So they produce little force, and it takes them longer to reach that force. But on the other hand, they are highly fatigue resistant. So you can use them continuously for hours and hours and hours, and they won't get tired. And because of that, they need high amounts of mitochondria. So that's exactly what they have. On the other extreme, however, you have the type 2B or X, which have really high force generation, but they're really resistant, or they're not resistant to fatigue. They tire very easily. So that can happen in minutes of exercise or movement. You feel that burning sensation that tends to come from these cells. But on the other hand, also the speed of force generation is much higher. So not only is the force that they can produce really high at maximum, they can get to that force at a really quick rate, but their mitochondria amount are quite low relative to the type 1. And then you've got the type 2A, which are kind of in the middle. And I've created content on this before, but type 2A muscle cells are very plastic, so they can move between more of a type 1 feel and function and a type 2BX feel and function based off of the type of training that you do. So what are the factors that allow you to be in a really relaxed state and then in the next instant be punching the hell out of a bag or a person, hopefully not, is the first factor is the type of muscle cells that are activated, right? So for the posture, you're going to be focused mainly on type 1. And if you're using all your might at once, then typically you would use a multitude of cells. So you'd be using your type 2 cells as well as your type 1 cells. So you're going to be recruiting everything you possibly can. Now, factor 2 is the number of muscle cells that are activated. So you're going to be activating for posture or for really basic things. You're going to be activating, let's say, 1,000 muscle cells. But if you're going to be using all of your might, you need to be using as many muscle cells as possible. So you're going to be activating as many muscle cells as possible. Now, on top of that, I should add that there are other ways that you can modulate that by the number of times that you activate a muscle cell as well. So if you were to activate, let's say, this muscle cell 50 times, as opposed to here, you activate it maybe 5 times because you don't need it to be activated constantly. You can have it be activated a lot more infrequently. So that's another factor as well. Okay, well, I think that's where I'm going to leave things for this lecture, this part of the lecture. If you ended up sticking it out live or after the fact when I release this, congrats. That's a real dedication to wanting to learn about muscle. I'm going to be covering the second part probably next week. So if you would like to get a more complete education, it's going to be covering, again, metabolism, as well as maybe I'll throw in that clinical mystery, depending on how interested people are. So let me know if you are. And with that, thanks for stopping by. And as always, I always appreciate you, and I'll catch you in the next one. Bye.